But Cosmart in this actually we are phenotyping, but many times we are actually failure. We cannot throughout the year still not able to phenotype things. Okay, so my topic is actually here. All many students here, so I think I am not only present of what's the feeling what we done, but giving some basics about the first one on to say. Okay, so my topics now that's why it's current understanding progress in research of Ustilaginoidea virens. Now it is actually threats to all rice growers. Why? So this causal organisms, you know, this Ustilaginoidea virens is actually glioma. This sorry, it is asexuals, and this sexualist is velocicular virus. And it is first recorded in from criminal belly of the same state by Cook in 1830, long back actually, almost more, around 200 years ago. This in Panjai kingdom and Skomycetus Panjai actually host is automatically rise, but with some weed also reported for Cosmart also multiply. So this is a very striking symptoms. It is easily visible, but visible only at the crop ripening stage. At the initial stage, when it just started infection, that time it is not visible in the daytime. Only the persons who are working on they can who feel can recognize okay it is infected because little bulging is happening, and that time it is very minute white smart mycelial structure was protruding from the veins. So initially it's a very small protruding mycelial structure. You slowly converted. It takes around 20-25 days to form the mature smart balls. And by the time its color is also changing, the most common color is actually yellow, orange yellow, and it is black to black. These are the most common color. And this starting from yellow, orange yellow, all are actually spot already spoke is formation is completed. Okay. So regarding these distributions, actually this distribution map is taken from CAPI. It is very old. It is 1982. Actually, the figure is there. Only this much pocket of the many countries have reported about Osman Panjai. And this dark red is the most area where this disease is frequently appears. So in India, this actually AI said one production oriented surveys going on every years, and the analysis of the around 40 years data was published in one bulletins from IIR also. They have not only published the for your reference, if you want, we can look through these bulletins. They are actually how the all major and some of the emerging diseases are spread during these 4-5 decades. Initially, this two decadal displaying is yes. This from there is actually this first decade including 80, 81 to 90, it was intensity and geographical speed was dramatically changed actually. And epidemic form was observed during 2020 to 2040. This epidemic found in some part of the state, Haryana, Jharkhand, Tamil Nadu, UP, and the high intensity observed in many states have not reading the names and low to moderate in another information. So almost more than 12-13 state was widely a state of this past month. And still now in many states it is not like that, but every year high intensities are here. But Osmart is we have read that it is have qualitative and both quantitative effect. In other species like say blight, bacterial blight, where quantity is the major factor, that is the actually make problem in the productivity and production. But how smart study has started. We also initially started from some quality aspect. What actually it has negative effect on quality. So if we go through this impact of this fast mark disease, we have this first one is the major one, that is we know that being lost, that is the effect on productions. Then the quality loss. The major we have also taken initiative to work on, and health hazards we have not started any work on that, but because on this aspect already many works has gone in outside country, and there is some good publications we measure. Many times we refer about this toxin by Mao et al. And we published in journalism. So for health hazards, it is for your information, it is a cytotoxin and both in phytotoxins also. And rice, though this is less susceptible to exhibited phytotoxic effects, when it is more than 1,000 microgram per ml is secreted by the fungi. So, and is the seed health, these aspects, many references available. And we also have tested some of the another released varieties. And also we have worked on some quality as well as some another popular varieties. Okay. So we also published one in Journal of Cereal Sciences and another in Journal of Agriculture Sciences about this quality and quantity related works. So here, this is the sample. This looks itself is the problems in the market when you go for procuring seeds actually, because it's when a lot of phosphate is preparing because tracings it actually mixed up with the seeds. Though it it is that it is not sequence disease, but it all has effect on its seed. Why I'm telling. That's another thing is when this phosphate appeared in the panicles, it has a lot of deep grade is significantly decreases in compared to the healthy panicles, and also subsequently chappy grade percentage is much higher percentage in compared to the healthy panicles. Here, for your information, we have taken the grading sampling. Grid-based sampling. So we have to, because we have taken this large area data, because this quality aspects, it is necessary for unbiased sampling is required. We are actually more than acre area. We are basically taking the data street plots for this quality aspect works. So here we have procuring 3x3 grid method, and there we have subsequently 1 square meter 5 random places as per statistical sampling rule. Then we taking at least one more patient that we treated as the disease panicles, whereas no one is present that we have taken healthy panicles. We have taken these healthy and disease panicles from the same sampling size, and we have to take separately taking the healthy seeds together assembles and disease panicles are separately taken together. These are act as the disease sample and others are healthy samples. And now we compare these disease seeds with the healthy seeds. Whatever data we are compared, we can set to the healthy seeds collected from healthy panicles. And seedling giver, subsequently seedling giver is also affected. Here was telling that seed, it is not seed transmitted disease, but it has effect on the when seed germinates. This spore also started germination, and it is a to some extent it also spread inside the seeds, and that actually that actually affecting the growth of the seedlings. So that's why seedling giver is lesser. Both fresh weight and dry weight basis in compared to the healthy grains. So these are the production related. Here actually we have taken the panel of the varieties. So already we have screened the inner release varieties also, and from there we have taken this susceptibles, moderately susceptible, moderate resistance, and one highly resistant varieties. Here it was observed that this seed risk, though it was only 0.012 by maximum 8.5% weight loss was observed in this 14 panels of the variety. So, regarding to cookie qualities, there is also significantly low in diseased cereals compared to the healthy cereals. The hulling, head rise recovery, kernel length after cooking space, speed and water uptake and disease. So, one point I just want to browse see that this water uptake, this parameter is very much essential, important parameter because that's why the where this effect also in head rise recovery. As a biochemist, they told that water uptake capacity, if it is significantly lower compared to healthy, then there is definitely there is a effect in head rise recovery. So that's maybe the cause as his water uptake capacity is significantly reducing compared to the healthy grain. That's why head rise recovery also affected significantly. So and nutritional quality. Nutritional quality is amylose. We know that amylose is very important cooking qualities because it is our taste is affected by the content of this amylose. This amylose content and total phenol significantly high, whereas antioxidant activity is significantly low. This is another aspect why we are highlighting that this fungi also affect the quality of the rice. So this is actually this is the first kind of report on quality aspects. Report and no others report we have not observed in any literatures. So we have even started work on why this protein quantity affects. So we have started work on this amino acid groupings and other some important parameters related to the quality of the rice. So next I want to highlight about the disease cycle. Actually, this is the part still we are not reached in on full sums that exactly full disease cycles we have not known. Because we know this is the Ascomycetes fungi and Starval fungi. It started with the acide, this primary inoculum, the acide drops on the during the flowering stages, and it entered with the during the pollination. When the pollen actually started germination, and through its germination, the acide ascospores also started germination, and through the intercellular regions, they entered inside the ovary. And then their multiplication started, mycelia first development, this conjugation started and produced food. And this chlamydospore actually is produced essentially chlamydospores, and thus chlamydospores again spread during the flowering period, and they also similar way through pollen enter. So this is the hypothesis and probable cause of the infections. But still it is one part coming later. So this fungi, you know, this is produces glutamate in a sexual mode and sclerosia, that is the resting spore. And these actually then spores produce this from this sclerosia in the seasons. And this chlamydospore are found in some of the perennial weeds, the Vegeteria, Macinaca, Canicums, and Ticarica. This is also report was available. However, till now, no evidence of presence for how chlamydospore are germinating at the field. This is the point. Still, we are in search of exactly how the disease cycle is completed. So there is three hypotheses is here. One is pathogen exclusively infect rice flowers via chlamydia. This was by wind currents and obligor venous plus at the time or before flowers. So it is another observation. We are also started one epidemiology as well. Another aspect is that it is the hypothetically, and we are in the lab culture, we are taking when the temperature is slightly down. Actually, on story, not a story, it is of the observation can say, you know that this disease is not new actually. Even before evolution, this disease was present and it was appeared in the lab basis. And if you all know the pathologists told us that it is called actually symptoms of goddess of wealth. Lakshmi we call it. Okay. So why it is called? Because that time our weather was something different from now actually. Now temperature is rising up also, and there is no steep this some year we are getting temperature down just upward 15 onwards. Some years we are getting at the end of the upward. The erratic kind of temperature fluctuation is observed. But 40, 50 years back, that time there was actually sequence that is just after October 8, this temperature was started down. So rice actually prefers during its harvesting period little cold, very sunny, and with small moisture is required. So this pathogen also required the same kind of weather condition. So that's why which year production is very good, that year this was not is happens. But now, because of this changing climate, this fungi is happening. So that's why now the common actually your recommendation is that if October onwards, just starting of the oil of the rice, if there is a little drizzling or heavy UV positions with lowering temperatures, then this is the time of the probable infections of this pathogen. So this actually prophylaxis basis of spraying fungicide is recommended in the area where it is endemic type of and the variety which is the susceptible type of. That means it is our observation so that this variety is having. This is for the farmers we generally now recommend because when this is separate, that is no use of any management. So here, but this is the simple hypothesis. But rejection, while it is possible that foliar inoculation is excessively artificial and thus poorly represent disease epidemic. Another is hypothesis is this pathogen is predicted to seed and soil one. So histological examination, prediction with host and cause and stems of naturally severely infected chemicals are analyzed with a light microscope and transmission electron microscope. Actually, just initially, I have feeling that why it is not simple. This pathogen spore adhered to the seed. When seed started germinating, spore also started germination. It infect. It mycelium start growing, but after some times it stops growing. Whatever other secondary metabolites are there or toxin is produced, that's only effect of the seedlings. So which is not actually like Ustilaginoidea, it does not like uses not up to where this actually growing with the plant actually and reach the inflorescence. But here it is not happen. Actually, no evidences were observed after some time when microscopic or transmission here was done. So this hypothesis is significant. So the latest one what I was talking about, this is the sexual cycles and asexual cycles. So this part is not still clear exactly how the total cycle is going on. So this is the simple functional process what I was telling. Already I explained how actually infection process has happened. So these are the some photographs. You know this is the backdrop and are infecting franchise and there is astronomically colonized other rice organs and other plants. It research findings says it is this paper actually statues. So we also tried to use statues samples. It's really good. This statues is but we have once procured and culture on it. It's really good in compared to potato sucrose what we use. But statues is a high, very costly. So we stop. So as the research findings that statues and the administrative sugar is very good in practically. Also implies glucose and sucrose as carbon source. And this finding indicates that the infection of virens is unique and distinct from other flower infecting pathogens. These are the some photographs. And this is another interesting things published by Islam et al. In microbiology. They are actually sequentially photographs how the pathogens actually colonize static infection and colonize inside the ovary. So these are the on the left side, these are the uninoculated one, and these are the inoculated one. Day wise, they have microscopically and they have analyzed it, and it is how protrusions and bulging was happening, and how ovary also when uninoculated and inoculated they have compared. So these are actually my own photographs. We actually successfully in 2016-17 we first actually cultured it. And here in the gravity is also showing how with the times it transformed from mycelial to monoidal stage and chlamydospore is producing. And even we also reported even in artificial culture also will sclerosia is formed. There is still one question mark is there because we know this sclerosia how in field condition, practical condition, other with the nature it is formed the way there is one question mark is still there how this artificial media how it's actually sclerosia formed. This is under process actually we are also in search of the reasons. And this is the standard artificial incubation procedure in the net house conditions. And we also have done one small chambers with not much, very much costly things. Only one small glass house with temperature with AC alternate two ACs there. We are using it alternately. But here for mass screening, large area is required. That's why side by side with field, we also screen in the net house. We are simply poly house and making screening. So that's why I'm telling that throughout the year it's not possible because during May the high so high temperature is so high. So that time we cannot come down the temperature to 26 to 27 degree centigrade. But otherwise, during this time, which is possible, and this is very simple process. First, we cultured these, then we in the liquid PS media we was culture it, then we sieve it. Then it will take the chlamydospore 2% sugar solution before at least 2 to 3 years before inoculations, we take this chlamydospore in the 2% sugar solution and keep in normal temperatures. And also, this chlamydospore will be clean one chlamydospore. So, not old chlamydospore can take for inoculations. It is very immature chlamydospore. Clean one chlamydospore, which is actually during our work we have seen it that this clean one chlamydospore is good for inoculations. And another thing is the standardization of the booking time. Is the late booking stage, just 7 to 8 days before emergence of the panicles, you have to inoculate two microliter solutions and also look that these spore concentrations should not be 2 into 10 to the power 5 10 to the 6. So, if the spore concentration will be high, then there will be actually primordial will be loss is ability. Instead of infection, that emergence will be failed actually. So, these are the some of the steps. So next we actually also needed that any management practices you know that diversity analysis is required. We need to booking of the isolates. So we in 2 phases we have worked on diversity. One on the in the first phase we have taken isolates from northeastern eastern India, only 7 state covered. And here also we have taken at least representative districts of the states. Not like that only the 4 5 state from one places. It is here with 63 isolate is the good this distribution is very good. At least representative districts and in some cases, if not, it is at least 2 blocks, separate blocks wise isolates are taken. And in another, we are taking from northern, it is actually Himachal Pradesh, Uttarakhand, Uttar Pradesh, Chhattisgarh, and Madhya Pradesh. This second project, we have done the diversity study. So there is some morphological study, and it was also reported that there is a lot of diversity in the growth of the fungi. It was observed that one group of fungi is spread fast, its mycelial growth is occurred very fast. But some isolates, their mycelial growth is not occurred in much faster rate. They simultaneously mycelial growth started to some extent, then started conjugation was started simultaneously subsequently. Whereas in some isolates, first huge growth is there, it's almost petrified within 15 days, and then it started the sporovations. So here this is some representative purpose. So here we have seen that around we have 79 isolates. There is we have schooling as part of the growth of the fungi, and it was observed that after 7 days, 74 isolates reach 10 millimeter growth, whereas 7 isolates only reach this. This is actually 14 days. This blue color is 7 days, and this orange color is 14 days, and this one is 21 days. So it was observed that this score 9 is actually more than 40 millimeter growth was observed only 20 isolates on 21 days. So there is also some mainly 3 major texture was observed in all these isolates. The compact, cottony type, and party type texture was observed. And this is a side view, and this is the overhead view. This first maximum was compact, and only few are cottony texture. And another thing is the color. Actually, color was somewhat correlated with the sporulation stages. Actually, so here we have taken this 4 glucans. Yellow is white, yellow, greenish yellow, and greenish black. Actually, okay. So this is the how it's changing. This is actually same date after 21 days. After 20 this observation after 21 days. At 21 days, this isolates has mycelium has only initiation. Part of this sporulation started in, whereas in this 76 number isolates is just sporulation stages. It's a part of the mature, almost reached to the mature, and here some of the subject formation of the spores here, parts of the species started in the dormant structures. And these are the dendrograms. Here is mainly 2 groups was observed. Cluster 1 is actually we have Meghalaya, Assam, Odisha, West Bengal. Did not show any clear segregation based on the geographical location. So it is not like that northeastern isolates for northeastern are grouped together, and this eastern region states are grouped together. Not like that. We are actually mix up is there. But still, there is found that it is actually higher. Is you can say that how is it possible? This Assam, West Bengal, Odisha, these are adjoining states. So we have observed which one actually grouped this Assam isolate with this one. We observed that this is actually the northern part of the West Bengal's isolates, or Assam isolates with the neighboring districts isolates actually with us. So, although it is not segregated state, but it is ecologically it was segregated. And in cluster 3 comes mostly from Odisha and West Bengal. They are actually just the middle part and the adjoining areas of the Odisha West Bengal parts. So I don't think it's necessary to explain. You know this diversity how we are explaining. And in northern Indian isolates, here actually we have got three major clusters. In cluster 1 covers mainly MP isolates, and cluster 2 comprises isolates from all states. Means all states means here actually from HP, Himachal Pradesh, Uttarakhand, and Uttar Pradesh also, and Tamil Nadu isolates are there. But cluster 3 comprises mainly from the HP. HP also again there is the elevation is there. If you go in the solar and the below also region, there also price going area is also there. In some of the upper parts, there also sir, no, this HP is high price condition. So actually that's why which cluster 2 has mostly UP isolates, and cluster 3 covers mainly much of this isolates. So here are those things not much times. And another thing is diversity. I mean, diversity is there. So we also thought diversity. Why it is diversity? So one major reasons maybe behind it is the as it is the sexual stage is there. So is there any mating type analysis? Different mating type analysis there. So we also analyzed the mating types. And it was we have used two primers, 22 primers, and this 74 isolates. It is mix up of the northeastern India isolates. Here we found that there is both homothallic and heterothallic isolates. Both are actually present. So it's maybe one of the major reasons of the diversity of this population. So the management aspects, though not much work was done, it is also it is the beginning of the strategy management of this project. So cultural practice, so what literature, some of the literatures was found that special pattern of natural space for the proximity of directions, and higher this rice grows near directions, there first town was much higher in compared to the other places. And another thing is these eastern regions, there is also deep and sensitive situation, rises calculation, deep and sensitive situation. So here also this severity of the housemark is much higher than the in compared to the irrigated areas rice calculations. What are the reasons? Possible reasons are we are talking about this is the much they need much more actually moisture. And what are the source of moisture in the deep and sensitive situations? Actually, what happens there is the dew deposition is comparatively higher than this irrigated areas. So that's maybe the possible regions of higher intensity of the smartphone appearance in the deep sensitive situation. So this was actually not repeated, but as I told, they also from Bangladesh they reported that the drainage channels are coming in place for climate conditions. So they advise to clean them properly. And another thing is the nitrogen loads also influence the disease occurrence. Like other diseases, this false part also prefer high heavy nitrogenous fertilizer used to feed. And it is actually observed that it is very simple experiment we have done. And there is a simple code is this heavy nitrogen fertilizers like other diseases, heavy nitrogen fertilizers also use spot ball formation. And Brooks et al. This is actually this American scientists, they also work on the crop rotation with soybeans. They found it is so we also worked on the staggered cultivations. We have taken month-wise sowing and then transplanting starting from June onwards. Though what its report is there is not exactly very much correlation, also strict correlation observed. Although this there is actually varietal response is more than the so flowering stage occurred in September to and October in both the years because of continental weather conditions available. So they are probability with this during best weather conditions. So here actually we have our aim was to track exactly which regions, which time, which temperature actually energized to form spore or infection of the starting. So we also together started, as you know, that this disease should detect at the very early stage of infections. So we also tried for detection procedure like other disease. Here we also tried for lab-based certifications, and we have successfully. But and it was already done, but not. We are in the process how it can be very cost effective and pharmaceutical. That's the challenging part. LAMP is for many diseases like other many virus diseases, bacterial viral disease, all diseases of them. But how it can be taken to the practically field and pharmaceutical, that is the challenging part. And we also worked on this latest technology that recombinant polymerase based detections. And here, in compared to the lab, this update is much better. And we are also in the process of this lateral flow assay based method. It can be, we can succeed. That will be actually the success of this works. And we have just initiated with this work, so this is not our way. This is some of the available reports what that the individuals are already identified, and we also have already panel is ready. This is actually our last 7 years. We have screened sequential screenings and repeat screening was done. Made the panels. We our start work was just started. So here some of the report which are the individuals are identified as leaders, and another is we also have identified two people. These are the some of the varieties also found. That's a list of the list we have just find out this. So some Chinese scientists also tried for this biological medicine. They thought they had from the substance of bacillus species solution of 2.5%. The organisms are found effective. Don't much rely on this because our observation is that particularly that fungi-based bioconformation like trichoderma and others low importance bears. But our observation, if you start work, definitely you try to see the scrapping the bioconformation like if you try with some trichoderma based diagnostic, you would just scrap the fungi from the crust of the Ustilaginoidea, this housemark fungi, and take it in good. Because my observation may be right or wrong, I don't know. But my observation that is revived begins. So that means this trichoderma based fungi is not at all work on this Ustilaginoidea. Though report is there, it is working very fine. So it is controversial. I think it is contradiction. I take contradiction in the reported results. And this they also again during 2014 they reported that 2.5% one this bacillus this suspension block very common. But chemical management, there is many chemicals. Yeah, all are actually indirect assessment of interest, which works, but the result was based on the indirect results, which just compared with the control plots and the infected plots. So I myself also tested this type of system based fungicide in the field conditions. Another person also reported this propiconazole, and I also tested Posite works roots and Peruconazole also, Patisi also working roots. But for Osmart, how much quantity of the active ingredient is required, and here is I found some report of the resistant aspects of the Osmart from house actually. So I started this bioassay of three major groups of the fungi. Sorry, two major groups. One is the triazole and propiconazole, and another is diazoxide strobins. So we found this very this whatever the active ingredient is in commercial formulations, that is actually based on I'm telling that is indirect assay method. As it works on the other major fungal diseases, so it is tested in the formulation the same dose, but this formulation actually this formulation may lead to the development of resistance. And startingly, we also was surprised this after we also first find out the LD50 values for this respective three fungicides. This azoxystrobin, propiconazole, and tebuconazole. Carbendazole also done. It is missing here. So these are this part is 0.08, 0.05. So that means if we make it 100 for commercial formulation, it still come under this is 8 microgram per liters. This bioassays found that two fungi mostly is okay for around this most of the isolates are within the range of the LD50 values. Or this propiconazole LD50 is 0.05. So this much all are nearby of the LD50 value. But we are surprised that two isolates are less sensitive to this LD50 range of values. They are actually LD50 is found that is within the range of 0.07 to 0.08, much higher than the 0.05 LD50 in Paris. So it should be actually screen whether these two isolates is really less sensitive to LD50. But the thing is not that propiconazole in case of azoxystrobin also we found that here also we found this for most probably one isolates we got less sensitive to this LDPP values. And in tebuconazole also, tebuconazole maybe the reasons this tebuconazole is much used fungicide in eastern and northeastern regions. This against some of the diseases. So this may be the reasons. There is some isolates. Around 3 isolates are found less sensitive at LD50. They are actually sensitive much higher. About the 0.03, they are sensitive to 0.036 to 0.04%. So bioassay should be done, and on the basis of this formulation, this private people to think about the formulations. So because that common formulation used for syphilis, blast, or other major diseases, that is at the higher end of the active ingredient. Of course, not as our this is, but this is not for the case of the one fungicide. All 3 different fungicides we have getting as much lower concentrations of active ingredients required to inhibit the pathogens. So these are some of our publication on phosphorus. Thank you. It means very